Okay, everybody. So let's talk about eukaryotes versus prokaryotes. So by now you should have done your reading, and we need to talk a little bit about that. So real simply, eukaryotes are cells like ours. There's a true nucleus. These are a more evolved cell than the prokaryotes. Prokaryotes are considered to be a little ancient, kind of antiquish as far as cell life goes. They're like ours. So eukaryotes have membrane-bound organelles. They divide by mitosis. But really, eukaryote means having a true nucleus, where prokaryote means not having a true nucleus or before the nucleus, before the kernel. So these tend to be like our cells. These guys are more like bacteria or archaic cells. So like their DNA just floats in with their regular stuff. There's nothing there. There is kind of like a DNA area, a nucleoid. It's kind of a nucleoid area. This is where the DNA is normally found. These guys divide by binary fusion instead of mitosis. So, and they tend to reproduce by cloning themselves. There are ways they can mix up their genetic material, but for the most part, they kind of just clone themselves. Okay, so let's kind of take a look at these and look at what the differences are. So if you're a distance learner, these are the cells that I had the groups A through D look at in class. So we have a eukaryotic cell here, and I'm going to blow those up, and we have prokaryotic cells here. And let me kind of show. So there's our eukaryotic cells. So just for showing you, here's a cell membrane right here, right? It goes all the way around. Oops, let me go back. And this would be our nucleus right here. Actually, so these are what are called excretory cells or endocrine cells, and their job is to basically put stuff out. And so these are most likely exocrine cells. From what I'm seeing, there is a lumen right here. So these are found like in your pancreas. They're and what you have here are actual large vacuoles into emptying their contents into this empty space or lumen. So very important. This would be the nucleus. If you notice, there's probably a vacuole being formed here. There's another vacuole here. Vacuole, vacuoles. So whatever this is, if this is a gallbladder, we could be producing fluids for digestion. If this is a pancreas, we could be producing insulin. So really important. So that's what that is. Okay, whereas here I have prokaryotic cells. Now, if you notice, you don't see details. So really, let me point them out to you. These are the little hair-like things. These are bacilli. So these are rod bacteria. I guess some of them could be spirochetes. This is from a slide that's supposed to be mixed bacteria. So that's what's going on here. So, but if you notice, you just see little lines. A few cells in class had cocci, and you could, if you really, really focused, you could see the little individual dots. And it looks like that might be what's going on here. But for the most part, you're just, you don't see a membrane, you don't see a nucleoid, you don't see anything. But there, I mean, if you look at that, here's one cell compared to one cell. Now, these are both at 400 times. These pictures were taken through the microscopes in our classroom. So this is kind of the tops of what a light microscope can do. Now, this is very utilized in practice. I myself would look at urine analysis. I could tell whether or not an animal had an infection. So you'd see lots of cocci or rods. You could, I could look at discharge or anything under the scope, or we could send it out and they could do the same thing. So you can get a lot of information from this. Even though there's not a lot of detail, this is what we can do. I mean, you cannot see this with your naked eye. So that's how that works. Okay. Now, on the other hand, these are electron micrographs, which means these are taken through an electron microscope. Wow, what a difference, right? Here's one of those bacillus or bacilli, and actually this one looks like it's getting ready to do binary fusion because it's starting to pinch in here, and it looks like I have 2 areas of new nucleoids. Yeah, forming septum. So yeah, that's what's going on here. Anyway, so this would be, if you notice, lots of granules. You can see a membrane, maybe even a cell wall if we got close enough, and some nucleate areas. In our eukaryotic cells, this is actually lung tissue, so it's a little different than ours. These, you can see the cilia. These are the epithelial cells. This is a columnar epithelial cell like what we saw. It's a little smaller, but you notice that the nucleus is all the way down here at the bottom. That's kind of how you tell. You could call them cuboidal too if you wanted to. Epithelial cells are recognized by their shape, by the way. They're squamish, which they look squished and flat. Columnar, which is what these were. Okay, see how they look long like a column? And then these are more cuboidal shapes. So that. Okay, so here's the cilia inside of the lung, used to catch things that are not supposed to go into your lung. Also, it increases surface area. This is the basement membrane. But notice that you can see just a little bit more detail than you could. And yet we could actually go smaller and more detailed, which you'll see later on in some of the materials for this week. But really for the purpose here, I just want you to see there was huge advances made by the electron microscope. We actually discovered the mitochondria because we could kind of see them. We could kind of figure out they were there, but it wasn't until the invention of the electron microscope that we could actually see things. And we're actually going to talk about some of that differences when we get to Davis and Denali membrane model. Okay, so but let's stick to what we're doing. So prokaryotic cells and eukaryotic cells differ in a number of ways, and you need to know this. So this needs to be in your notes along with what the electron microscopes allow us to do. But you need to be able to tell what the differences are and be able to compare and contrast these. So this should be in your notes. If you've already done it in your reading, that's fine. If you want to add to it, that's fine. Or if you haven't done the reading yet, get the notes done now, however you want to do it. But it should be in your notes. I'm sorry. So it's different in the way it's DNA. It's different in the organelles. It's different in the way it reproduces, and it's different in the average size. Now, there are some exceptions to everything, just like in the cell theory. Okay. One way to remember this is DORA. Okay. So D-O-R-A. That's just what it spells. So DNA. DNA in prokaryotes is naked. It doesn't have a membrane that protects it. DNA is circular, and you don't really need to worry about introns right now. You could pick those up next year. Whereas in eukaryotes, they're bound to a protein, they're linear, they're inside a membrane. So organelles, well, we don't really have a true nucleus. We do have a nucleus in eukaryotes. Prokaryotes, no membrane bound. Here they are membrane bound. Ribosomes, they are 70S ribosomes. This is the size in prokaryotes. So this is smaller than when a eukaryote, which is 80S ribosomes. Okay. They reproduce by binary fission, and then these reproduce by mitosis. Unless it's the sex cells, then it's meiosis. These guys have a single chromosome, and these guys have paired chromosomes. And whoops, last but not least is size. Eukaryotes are always larger. Well, with an exception. Eukaryotes are normally larger than prokaryotes. So if you notice, if we go back, there's our prokaryotes, there's our eukaryotes. Much difference, same magnification. These are both at 400 times. So a much smaller cell here than here. Okay? So like I said, this needs to be in your notes. You need to be able to tell the differences between the two. All right. Thanks, guys.